Hey everybody, we're in the last section of chapter 3 and this is all about cell division, and the type of cell division we're talking about is mitosis. Mitosis is division of your somatic or your body cells, and the function is to produce two identical cells from one parent cell. Because we're doing this, we have to make sure that we double the amount of DNA, so we replicate all of our chromosomes. And we increase the number of organelles so that each cell will have those organelles. Each cell will have their own nucleus and a complete copy of genetic information. There are two phases in mitosis. In cell division, there are two major phases. Interphase, which is the longest period in which the cell is preparing for mitosis. And then we have the mitotic phase, which is a very short period. I just thought this was a cute cartoon. Love it. Hopefully you found it amusing as well. So when chromosomes actually get ready to undergo mitosis or go through mitosis, they're going to condense. So initially you have that chromatin, and when it condenses up, it looks like that X-shaped chromosome that most people know. What you're seeing here are two homologs. So these are one chromosome from mom, one chromosome from dad. They're identical to each other in size, in shape, and in genetic information. The difference really is the alleles that are on each chromosome may vary. So this chromosome may have alleles for blue eyes, and this chromosome may have alleles for brown eyes, or long fingers and short fingers, or insulin production and diabetes. Who knows? That's the difference. So they are identical otherwise. They have the same centromere positioning, and the structure, the kinetochore, is what actually binds the two chromosomes, or the two sister chromatids. So this is one sister chromatid, this is one sister chromatid that's already replicated. They bind them together so that you end up having these X-shaped chromosomes. So cell division has that interphase period, and then you have the mitotic phase. So all of this gray material here is interphase, and it's separated into three series, or three regions. We have the G phase, the S phase, and the G2 phase, the G1, S, G2. Some cells enter a G0 phase where they are not dividing at all. They're just kind of in a rest phase. In general, most of our cells don't do this. There are some examples, including female sex cells, which we'll talk about when we get into the reproductive system. And so we'll go through each of these phases and what's going on, and then we'll go into the mitotic phase. So in G1, chromosomes are getting ready to be synthesized. We're producing proteins for DNA synthesis. We're also starting to enlarge the cell. In the S phase, this is the synthesis phase, DNA replication occurs. And then we go into G2 phase. G2 phase is where we produce proteins for mitosis, but we're also going to prepare the cell for division by increasing the size of the cell some more and making sure that all of the organelles have doubled in numbers. And then we enter that mitotic phase. So here you can see interphase at G2. So we've already doubled our DNA. This is a picture from Fotolia.com. I found it on Google Images. Again, I love Google Images. And I just like the way they put it together. And your textbook, which I'll show that image next, has a little more information written down about the different phases, but I like this one because it just shows it very simply. So we go into prophase. Prophase is the first step of mitosis. During prophase, chromosomes are going to condense, and spindle fibers are going to form from these centrioles. So centrioles move to the opposite poles, spindle fibers form, and they're going to attach at the centromeres. Okay? To those kinetochores. Once all the spindle fibers have attached, then they're going to move the chromosomes into a line on an imaginary plate called the metaphyseal plate. And this is termed metaphase. So now we have all the chromosomes lined up. Each chromosome consists of 2 sister chromatids. So during anaphase, the chromosomes are pulled apart. The sister chromatids are pulled apart so that one of the chromosomes moves to one end, the other goes to the other end. At the same time, you can start seeing that the cell is invaginating on each end, so it's pushing in. This is the start of cytokinesis. In telophase, chromosomal membrane reforms around the chromosomes on each side, and cytokinesis continues until you end up having 2 identical daughter cells from one parent cell. So this is your figure that shows the same information. Here we have prophase. The difference is the pictures are a little smaller, so that's why I had the other one. Chromosomes condense, spindle fibers form, nuclear envelope breaks down. The spindle fibers attach to the centromeres at the kinetochores. And now they're going to start moving the chromosomes around, bringing you to metaphase. Once all the chromosomes are lined up, one on top of another, then you're at that metaphyseal plate, and then the spindle fibers will pull the sister chromatids apart. That's anaphase. At the same time, you're going to start cytokinesis, where actin proteins are going to start invaginating on each end of that cell. And then we end up with telophase, where new nuclear envelope reforms around the DNA. DNA uncondenses, and the spindle fibers come off of the chromosomes. Cytokinesis continues until we end up having 2 identical daughter cells from the one parent cell. This is an animation of mitosis.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between 2 daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attach to the kinetochores of each chromosome, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds. Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into 2 daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
So you don't have to worry about plant cells. Obviously, we're dealing with humans, and humans are animals. So the cell cycle has to work properly every single time, and if it doesn't, then you can end up with cells that have not the proper organelles, or not the proper proteins, or not the proper amount of DNA. And so we have certain proteins in our cells that act as controllers of the cell cycle. Cyclin and CDKs are your proteins that they watch the cell cycle and they make sure everything is working properly. And so there are 3 different checkpoints that your cells go through to make sure that the cell cycle is running properly. So the first checkpoint right here, you're going to make sure that proteins for DNA synthesis have been produced. We're also going to make sure that the cell is getting large enough so that the mitosis can occur. Then we're going to go through DNA synthesis, and we're going to hit the G2 checkpoint. In G2, we're going to check to make sure that we have all of the proteins for mitosis produced, but we're also going to make sure that the DNA that was synthesized looks correct. So we don't want any mutations to occur, because mutations can lead to diseases, cancers, and that's not good for our cells. We then have a checkpoint in mitosis at the metaphyseal plate. So at the M checkpoint, the mitotic checkpoint, we're going to be making sure that the DNA is lined up properly, that sister chromatids are attached to each other, and that chromosomes are lined up one on top of another, going all the way down that metaphyseal plate. So that when we do rip those chromosomes apart, they are pulled one sister chromatid on one side, one sister chromatid on the other. So each of the new cells will have identical copies of the DNA. Any of these checkpoints does not produce the proper results. So if at the end of, or if at this checkpoint we don't have the proteins produced that we're supposed to, or if the DNA is mutated, or if the chromosomes aren't lined up properly, that's going to, the CDKs and the cyclins are going to signal cell death. And that's termed apoptosis, programmed cell death. So that instead of having one cell start growing out of control because now it has no ability to control itself, maybe the chromosomes are wrong and other things are wrong, the cell dies and a new cell can be produced. So there's also an animation from that same website. I like this website, you can tell, on the control of the cell cycle. There's also a game out there, which I think is fun.
The process of cell growth and division in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed, and the cell will enter the synthesis S phase. During the S phase, DNA replication occurs, so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and two daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
So that's your cell cycle. The last thing we're going to talk about then are how cells become what they are. So the initial cell that was formed when or during fertilization was the zygote. The zygote is a single cell that contains a complete copy of genetic information, and this cell undergoes multiple mitotic divisions. So initially, it is a single cell. Within the first 24 hours, it becomes a double cell. So there's two cells, and then 4, and then 16, and then it just keeps getting bigger and bigger and bigger. So once the cell has hit the blastocyst stage, it's going to embed in the uterus. And that's about day 3 or 4 that the cell will actually, or the ball of cells will actually be able to enter and embed in the uterine lining. It takes approximately 3 weeks for the embryonic disc to form. So by the end of 3 weeks, you have an embryo. An embryo is a structure that is composed of 3 different germ layers: an ectoderm, which is over here, mesoderm, and endoderm. Initially, or prior to that, all of the cells are just embryonic, or are not embryonic, are totipotent stem cells. A stem cell is a cell that is able to kind of choose whatever path it wants to. So it's kind of like a college student that hasn't chosen their job yet. They know that they want to go they want to maybe they want to go to college, but they don't know if they want to be a law enforcement or if they want to go into banking, business, or nursing. Once the embryonic disc has formed, we have these cells here, right? And each of these cells is more specialized. So it still is a stem cell, but it now has only a certain number of options. So in that case, it would be a student that maybe has chose to go into that medical field, but they don't know if they want to be a radiologist, if they want to go into nursing, maybe they want to go into physical therapy, maybe they want to become a doctor. So they've chosen their line, but they can still become a lot of different things. So then you have multipotent stem cells, which are stem cells that will produce new copies of structures. So like you might have epithelial stem cell that produces more skin tissue or epithelial tissue. That would be an example of that. And so a totipotent stem cell is one that can become anything it wants. Pluripotent stem cells, they can become anything within a designated line. And then you have the multipotent stem cells, which are stem cells that produce new copies of the cells. And so this is a horrible picture. I just noticed how bad it is. But up here you have this multipotent stem cell, that can produce any type of blood. So it can only produce blood, but it can produce a lot of different types of blood. And so here it can differentiate into a myeloid or a lymphoid stem cell. The lymphoid stem cells can produce all of your lymphocytes. Or you can move over here and produce your other white blood cells or erythrocytes. That's all I have for you. This is the end of this chapter, so I'm going to close this out and upload this to YouTube. At this point, you should be able to answer all the questions at the end of your chapter. Now, this chapter I've decided to do in 2 parts. So the first half is going to be covered in the first exam. The second half of this chapter is covered in the second exam because it's a long chapter, like uber long. So I'm just cutting it in half, make it a little bit easier for you, and hopefully you'll be able to focus better. All right, bye.